This section is going to cover prokaryotic and eukaryotic cells. We will cover eukaryotic first, and I'll just give you a summary. This is written out sort of long style because this is an online course, and rather than just be maybe an outline, it is written out verbatim. But I'm going to go through and talk about each organelle, try to keep this short. So eukaryotic cells mean a cell with a nucleus. A prokaryotic cell is the term prokaryotic means before nucleus. This means true nucleus. So I'll do another video on this one. We'll do eukaryotic cells first. So starting here first, nucleus, nuclear membrane. In your PowerPoint, you have these pictures, and then in the lab lesson, when you click on that, you'll see this big picture here. So I'm going to use this picture to start with. So see that area in the middle? That's the nucleus. The dense area that's darker there is supposed to represent the nucleolus. And then this area around that is called the nuclear envelope. And it's got two layers. It's a selectively permeable membrane. It allows certain things to cross into the nucleus, and it's going to exclude others. And if you get close, you can see there are some lines inside the nucleus. Those are chromatin fibers, and those are long strands of DNA. They condense when a cell is dividing. In this case, this cell during mitosis, they will condense and form chromosomes. So the nucleus controls all cell activities. The reason is that's where the DNA is found. So yes, it's spherical, overall function, controls all cell activities. And that's because the DNA is found inside there. So it's called the control center of the cell. The nucleolus, I showed you that little dense area. It actually also has a function. It's called a site of ribosome formation. We haven't talked about ribosomes yet. Those little dots down here and the ones up here that are attached, the little purple dots, those are ribosomes. They are involved with protein synthesis, and we'll get to those later. But that's the nucleolus, is that dense area that's found in there, and that's what that does. That little dense spot, and then this was the nuclear envelope, just like it says right there. It's a double layer of phospholipids, and we'll get to that in a minute. Okay, now the membrane-bound organelles. These are not found in a prokaryotic cell, but they are found in a eukaryotic cell. So here is all the information. I will go through and give you a summary of everything. And there's the nucleus. Now all of this up here, all of that, including this, is all called the endoplasmic reticulum. Cool words. It's a series of elongated tubes and fluid-filled vesicles. The parts that have ribosomes attached, and you can see the little dots up here, that's called the rough endoplasmic reticulum. Rough because it looks like it's got a speckled appearance. This down here, see the little tubes sticking up that do not have ribosomes? That's the smooth endoplasmic reticulum. Those lack ribosomes. But what this does overall, this transports substances around the cell interior. And the rough endoplasmic reticulum has a side function of protein synthesis, and then the smooth has a little side function of lipid synthesis. But everything in a cell is highly organized. Nothing just floats around, and this is a transport. It's like a subway system within the cell. And I could probably go down. There's a nucleus. And these lessons are great. So there it is. So here's the rough endoplasmic reticulum, and then there's the smooth. Okay, a Golgi apparatus, or a Golgi complex. This guy, Mr. Golgi, scientist. Now, that's his Golgi body, Golgi complex. Sometimes you'll see it called a Golgi apparatus. Got a lot of different names. This is like the gift wrapper at Christmas. Now you can see it here. So on our big cell, there it is. It's a half a dozen flattened sacs. Each sac is called a cisternae. And what these do, now these things coming off here are vesicles. So, and you can see it's attached to the endoplasmic reticulum. Anything the cell produces, it has to go through the Golgi apparatus to be secreted out of the cell. So the function for this is it modifies and wraps up substances to be secreted out of the cell. So it packages it up in that little vesicle, and then it's released to the outside. Yeah, there's some microvilli, some cilia, but that's what it's like the gift wrapper at Christmas. A Golgi body is going to package and modify a substance to be secreted out of the cell. Mitochondria. Oop, wrong tab. See these little half-looking Tylenol things? That's a mitochondria. They are, if you cross-section one like that, you see it's got the folds inside. What these do, they're very important. They're called the powerhouse of the cell. Those take food energy and they convert ATP using oxygen. And the whole process occurs along this pathway. We'll talk about that a little more later. It's called the Krebs cycle and the electron transport chain. And the electron transport chain occurs inside of the mitochondria. So does the Krebs cycle. But anyway, that's they're powerhouses. They make energy in ATP. Lysosomes. So these here, and this is actually a good diagram as well. Lysosomes are very important in microbiology. Lysosomes are little organelles within a cell, and that's what we're covering here. Organelles are like little organs, and they're only found in eukaryotic cells. Prokaryotic cells are very simple. They do not have membrane-bound organelles. But if you go back up to our bigger picture, and I'm using this one because it's just a larger view. But down here, and you can see these the purple, those are ribosomes. There's a microtubule here, and this is a vesicle, but some of these solid, they're spherical looking structures. There's one right here. Those are lysosomes. And then there's another kind as well. It's called a peroxisome, but most of those are lysosomes. And this is kind of showing the digestive enzymes. What lysosomes do is they break down a substance, either food particles, worn cell parts, or microbes that get inside of a cell. And this is what this is showing. Look, the cell has brought some food across. Here's a lysosome. It goes up and attaches here, and we're going to talk about that later. This is called a phagolysosome complex. But it breaks down those substances. So very strong digestive enzymes, and they break down a substance within a cell. Vacuoles and vesicles, really just need to stick with the term vesicle. Maybe I'll just go here. I think I have... Anyway, so let me see. So a vesicle is this. And see, here's a Golgi apparatus. So vesicles coming off, and vesicles also, you can see up here at the top, it's where it's pinching in and it's forming a vesicle that's bringing something from the outside of a cell. So what they do is they move substances inside or outside of cells. Microtubules, and I'm going to go back up to our bigger picture. I hate to keep zooming back and forth. But here are some lines in here. Those are microtubules or microfilaments. They actually support the inside of a cell. It's called a cytoskeleton. And they give a cell some rigid support. Can you imagine as a human being trying to move around if you didn't have bones? That's kind of what a cytoskeleton does. So they're made of a protein called tubulin. And like it says there, they provide cell support for the cell flagella and cilia. And I didn't point that out. These are cilia. See the little purple tubes that are inside? Here is a flagella. See the tail that's coming off? These over here are called microvilli. Don't worry about these. But yeah, cilia, they're important. They move a substance across the cell surface. And then here's flagella. Ribosomes. I just touched on those a little bit. Ribosomes are these little dots, and they're also attached here to the dots that are attached to the rough endoplasmic reticulum. They are protein factories. They help make enzymes, cell parts. They make all kinds of different things, and the they're very important to a cell. In fact, there's a lot of drugs, antibiotics, modified antibiotics, that are called ribosomal agents, and they take out ribosomes because bacteria cells also have ribosomes. So small spherical organelles contain enzymes capable of linking amino acids together. So they essentially will synthesize proteins, and we'll talk about those more detailed later. Cytoplasm, and we're going to be wrapping up this first video, all of the jelly material that's within this cell is called cytoplasm. It's made of a protein called cytosol, and it helps hold all these little organelles in place. All the biochemistry that occurs within the cell happens within the cytoplasm, so it's got amino acids and things the cell needs, and it also helps give the cell some shape. All right, last part to this, cell membrane. Now, I'm hoping on here, and I should have, and it looks like these are the microtubules I was telling you about. There we go. This is a cell membrane. So basically, the most exterior part of a cell. It's two layers of what are called phospholipids. The little heads are the circular parts on the top, the tails that come down like this. Hydrophobic tails, that means that they are water-hating. This is a hydrophilic head, water-loving. This means that the heads are dissoluble in water and the tails are not. Fatty acids are not dissoluble. They're not soluble in water. If you cover a pan with fat and then put water on top, the water beads up. So that structure, and then all of these things you see sticking up, these are proteins and channels and things like that. This allows this membrane to be what's called a selectively permeable membrane. It allows certain things to cross over, and then it's going to exclude others. It's just like the guy at the club that lets, the bouncer, that it's going to allow certain individuals into the club, and it's going to tell the other individuals you can't go in. That's what the cell membrane does, and it's that very outside layer. Okay, that's the end of our eukaryotic cell overview.